Well, thanks so much, everyone, for joining. I really, it's so great to see people here live. And if you're listening to the recording, very happy to have you registered and listening to the recording as well. So like Angelica said, we've run this actually in the fall. So this is a repeating series, but really, I think it's something that there's a lot of information and content in the 4 webinars that are part of the foundation series. And so it's really helpful, I think, to re-listen to them if you have the time and are able to, because every time you listen to them, you maybe have seen a patient or a client that you're like, oh yeah, that resonates differently with me now because I saw this person. Or maybe it's someone that you're struggling with and you re-listen and you think, oh, okay, like this is something I can take home and utilize and implement with them. And so, so today what we'll be doing is we're going to talk about the structure of the intestines. We're going to talk about, kind of get a deep dive of the various cells within our GI tract. We're going to talk about the gut immune system, the GALT, which we are all probably pretty familiar with. Do a very brief overview of the gut-brain axis, but then we're going to go through the digestive process. And I do have a few stars that are on a few of the slides. And whenever you see a star, that's a slide or a concept that we are going to be building upon in future webinars. So our goal is that you're doing these in sequential order so that you can build on your knowledge. So the goal today is not to feel like you're back in anatomy and physiology from your college or university days, although it may seem that way as we're going through the content. The goal is not to overwhelm. There will be quite a bit of content, but like Angelica said, just take and absorb. I'm going to try throughout the presentation to help connect this to the why of why we're talking about this concept, why we're going back into a deep dive on these little, what seems to be nitty gritty. And the reason for that is because if we're building a house, if you as a clinician are trying to really work on root cause, figuring out what's going on with your patient, how to best intervene with your client, and then also just really feel like you have a good, solid foundation for your interventions. We have to build that foundation through understanding what's happening at the teeny tiniest little level, because what's happening at the cellular level is going to affect what's happening in clinical presentation with your patients. So, the goal here is to say, let's build this really solid foundation so that the house that you're building around your clinical practice is really firm. Because if you have a house that sits on a cliff, you want to make sure you have a good foundation. And we all know that there will be those patients who sometimes stretch us, or they challenge us, or they put us into a corner. And we think, well, how am I going to work with this patient? And if you can go back to those foundational concepts, and if those are really strong, then you're going to be able to withstand and work through that patient more easily. So that's really the goal here for today, and really for the next few webinars as part of the foundation series. So let's start by talking about some intestinal anatomy and physiology with just some basics here. So just super basic. We are built out of cells, which turn into tissues, which then those tissues come together to form organs, which then the organs come together to form organ systems. And those systems are working together to essentially help us to function as a human being. So they regulate different biological materials, they facilitate the elimination of waste and detoxification, they maintain things like body temperature and pH and other conditions that are optimal for different cellular processes. And when we're talking about the GI tract and digestive system, from a functional standpoint, it's really about digesting food, assimilating and absorbing those nutrients, and then helping to eliminate solid waste. Now, we're going to talk a lot more about the immune system and some of the other things that are part of the digestive tract, but those are the main 3 functions that they have. And then, like I said, we're going to go through the organs of the digestive system in sequential order. And the reason I'm doing that today is because, again, when you're thinking about what's happening in the colon, when you're looking at things like dysbiosis, and Kara is going to be talking about the microbial structure and function in the next foundation webinar, we have to know what's happening above the colon, what's happening above the digestive system from a top-down approach to understand why things are happening further down, because that's how our body is made. We eat food, it goes through the upper part of our digestive system, and then it goes into the lower. So understanding each of those different pieces will help you to better connect those dots. So that's why we're going to go through that again today. So let's just talk about the importance of an epithelium. So at the very basic definition, an epithelium is just a sheet of cells that covers a body surface or lines a body cavity. So we have the intestinal epithelium, we have a lung epithelium, we have a GI or a gastric stomach epithelium. So it's just this sheet of cells that are essentially covering that surface or they're lining a cavity, and it forms boundaries. And that's really important, especially when we talk about the intestinal epithelium, because that's basically that boundary that serves as protection. It helps facilitate absorption. It helps to filter things. Think about the kidney epithelium. The epithelium of the kidneys has a certain role and function. Excretion, sensory reception. And in all epithelial linings, they're bound together by tight junctions. So we oftentimes think about tight junctions in terms of the intestinal epithelium, but there are tight junctions in other epithelial sheets of cells in other body areas. And as long, we're thinking about the intestines, as long as the contents that we're ingesting are in the lumen, which is, if you look here, it's this innermost part, it's that hollow central part of the tube of our intestines, it is still technically outside of the body because we have an opening of the mouth, we have an opening at the anus, and if things are going all the way through, they technically are outside the body. So this is why when we have a strong epithelial layer that's keeping things in check and it's keeping what's supposed to stay in the epithelium or in the lumen, if it's keeping those in the lumen and they're not getting into the bloodstream if they're not supposed to, that's when things are in balance. It's when we start to have this breakdown and things from the lumen are then getting into circulation that then triggers that inflammatory response. So we'll talk a little bit more about the lumen and some of that in a little bit later in the presentation. And again, the lumen is where we have all of our commensals and occasionally pathogens. It's where our microbiome resides, is in that luminal area, as well as whatever they're giving off. So whether it be lipopolysaccharides, certain bacterial byproducts, all of that's going to be contained in the lumen as long as we have a nice epithelial barrier. So if we break it down, and we'll first talk about the intestinal tract regarding the small intestine. So there are 4 layers to the small intestine. We have the mucosa, which is this, this kind of larger area here. And that does include this epithelial cells. We're going to talk on the next slide more about the epithelial cells individually. And again, the main function for this mucosa, this kind of first part of the small intestine, is to really absorb and transport those nutrients and to protect the body from pathogens. We're going to talk about the Peyer's patches and some of the immune barrier systems that keep that in check. There's then a submucosa, which is a thinner layer. It's very collagen rich. And that's going to support the mucosa, so it's right underneath it. But it kind of connects that mucosal layer with the muscle layer. And that muscle layer is really more about that movement. So when we think about peristalsis, propelling food through the digestive tract, but also the migrating motor complex, which is, if you're not familiar with the migrating motor complex, it is a very important concept related to SIBO, because this is our motility that happens when we are fasting. It is only active during a fasted state, so between meals or overnight when we are sleeping. Once you eat a meal or you eat anything that has any nutritional value, that migrating motor complex shuts off and you flip back into peristalsis. And the migrating motor complex is really just this sort of cleanup system. It's like taking a broom in between your meals. And let's say you have young children and or toddlers who have thrown a bunch of food on the floor. You want to come in between those meals and clean up and sweep that through before the next meal, because otherwise things build up. And so that's what the migrating motor complex does, is it helps to kind of clear things. And so that's all happening in this muscular layer beneath the submucosa. And then we have the adventitia, which is a smooth muscle membrane that has 2 layers, and it's mostly secreting various fluids. So going to this epithelial layer that's part of the mucosa. So that's that single cell layer that we talk a lot about when we're talking about leaky gut or intestinal barrier permeability. It is made up of enterocytes. Enterocytes are just your intestinal cells. And they have there's several different individual types of cells that make up this epithelium. We have the enterocytes there, and then we have the Paneth cells, which these are only in the small intestine. So you won't find these in the colon, but they are making and secreting antimicrobial proteins. So we have in our epithelial layer our own inherent cells that are responsible for secreting antimicrobial proteins. So if these are functioning properly, this is part of that process of helping to protect us from any foreign invader or any dysbiotic pathogen that might be coming in. So these are in the small intestine. We also have goblet cells, which are responsible for secreting mucus. Now, I'll show you in a little bit, there's a difference between the number of goblet cells in the small intestine versus the number of goblet cells that are in the colon. And that has to do with the amount of mucus that needs to be secreted for protection. And so there are some goblet cells in the small intestine, but we have a much, much larger number of goblet cells in the colon. And then we have neuroendocrine cells. And what these cells do is they essentially are managing the nervous system response. So they're initiating digestive secretions, protective responses. They're kind of coordinating all of those neurological factors. We have a few other cell types in the small intestine. We do have some exocrine cells. So these are like the goblet cells would be a type of exocrine cell, but we also have cells that are secreting your various digestive enzymes. We have endocrine cells, which are responsible for secreting digestive hormones. We'll talk a little bit about CCK and secretin to help with coordinating things like bile flow and pancreatic secretions. And the more secretions are really, they're regulated by chyme, which if we remember from our biology days, our anatomy and physiology days from school, chyme is that partially digested food stuff, and it's typically mixed with a bunch of digestive juices. And so when that chyme starts to make its way through the GI tract and into the small intestine, that's going to trigger the signals for a lot of these cells to start secreting their excretions. So again, if we think about, okay, what if we have somebody who has a really inflamed small intestine? What if they have something like, celiac or Crohn's or SIBO, or some sort of condition, or maybe they've had parts of their intestine removed, if they are missing or, certain pieces of the intestine, or if they have an inflammatory condition that is preventing maybe some of these cells from working as efficiently as they should, that's when we're going to start getting some breakdown of mucus or maybe digestive enzyme secretion. That's something that happens in SIBO a lot. So if you are working with SIBO, we know that part of the pathophysiology of SIBO is that it interferes with these exocrine cells in the small intestine. And so then we get this decrease in these enzymatic output. And so we can have more of an issue with actually breaking down and assimilating our food, which is what the digestive system, one of its main roles. So kind of tying that into what happens when these systems break down. So here just, I think, again, I like pictures, and this is a image for you just to help with kind of breaking out the differences between the stomach and what's the different types of cells in the stomach versus what's in the small intestine in terms of anatomy, and then also the colon. So you'll see here that there's a few differences. One, the small intestine, because of its main role in absorbing nutrients, it has these villi. So you're going to see these more, higher finger-like projections in the small intestine, versus in the colon, we have more of the crypts. We're not going to see those big finger-like projections in the colon. The other thing that you'll notice in the small intestine are these Peyer's patches. We're going to talk about the Peyer's patches because they are the main immune system component of the small intestine, and really of the digestive system, are these Peyer's patches, and they're only in the small intestine. The Paneth cells, as I mentioned, which secrete the antimicrobial peptides, these are also only in the small intestine. And then we have these M cells, or these microfold cells. These are also only in the small intestine. So what's interesting, and what we'll talk about, is there's a lot more immune system support in the small intestine than there is in the colon. There's a reason for that. And we'll talk about that in a little bit. But here in the colon, we can see that the mucus layer is very different. We actually have two layers of mucus in the colon. We have an outer and an inner layer. So that's another anatomical difference. And so when we look at the colon, here we have that mucus. So in the colon, having that inner and outer layer versus the small intestine, which is this much, much thinner layer, and it's not attached to the epithelium, and it does have some bacteria in it. But with the colon, you actually have a layer of mucus, this inner layer, that is completely free from bacteria. And it is the only part, basically, that we know of to date of your entire body that does not have any bacteria. Because we know now that your urine can have a little bit of bacteria, your blood can have a little bit of bacteria, the vaginal tract, the uterus, which we once thought were sterile, those have bacteria. So all these different secretions and areas that we once thought were sterile actually have bacteria in them, and they have a little bit of a microbiome. But this one layer right here, this little teeny tiny space that separates essentially where you have the most bacteria in your colon, because that's the largest concentration of bacteria is in your colon, separating it from the basically, this whole sequence of immune cells that we have, it doesn't have any bacteria. And so this is why when we talk about, and if you're familiar with Microbiome Labs, we talk a lot about the mucus barrier and making sure that that barrier is very much intact, because if it's not, then things can start to break down. Now, there is an outer mucus layer that does have some intestinal bacteria, but it is still considered a mucus layer, but it's this inner layer that is the sterile layer. So just as a recap, and feel free, these slides will be sent out to you. You'll have them accessible to you. So have these as part of a binder, have them as part of a folder on your computer so that you can refer back to them. But just as a quick recap, our small intestines, where we do have most of our nutrient digestion, we have absorption, and we have immune balance. Whereas the large intestine is mostly geared towards absorbing water, electrolytes, and this vast concentration of bacteria. Now, in the mucus layer, again, single layer with some bacteria in the small intestine, it's much, much thinner. Whereas in the large intestine, it's very, very thick. We have two layers, one with bacteria and then one that should be sterile. In the different cell types, we have some goblet cells, more immune cells, and we have secretion of enzymes. In the colon, we have many goblet cells, and we don't have any cells that secrete enzymes. And then again, the difference between the villi versus the crypts. So why am I spending so much time talking about and referring to the immune cells in the small intestine and the differences between the small intestine versus the colon? That's because if we look here, I think this is a great graphic that shows exactly how that differs. Is if we think about the purpose of each part of the intestine. So in the colon, the immune system is really geared towards focusing on just dealing with and handling the vast numbers of commensal microbes that are there. So there's not really a ton of, and by the time it's gotten to the colon, it should have already gone through several series of antimicrobial checkpoints before it gets to the colon. And food should have already been dealt with and absorbed by the time it gets to the colon. So the colon is really more about how do we handle and just make sure that we keep all these microbes in check and that we're not freaking out every time we see a microbe, because it's going to be potentially coming into contact with many. The small intestine, however, the immune system is really focused there because it has to distinguish between what is a harmful pathogen coming in versus a food that we may actually want to absorb and assimilate. And the amount of bacteria is significantly smaller than what's in the colon. We shouldn't have a lot of bacteria, an exorbitant amount of bacteria in the small intestine, unless you have SIBO, in which that's a whole nother conversation. And that's why SIBO can be so disruptive to the immune system, because it's throwing off this whole balance of the immune system and the microbial population. So if we look here, we can see that in the up in the duodenum, which is that first part of the small intestine, we have a much higher secretory IgA titer. So we have a lot more secretory IgA, which is basically there to help with managing those pathogens and kind of regulating what gets presented to the immune system. We also have a lot more of the T helper 17 cells, which again, very protective as long as there's balance. When there's imbalance, that's when we get into problems with autoimmunity and inflammation. And then again, the antimicrobial titer is much greater in the small intestine than in the colon. So again, we have a lot more of the immune system factors like the pyrus factors and Paneth cells in the small intestine because of the job of what the small intestine is intended to do, with basically distinguishing between friend and foe as we're consuming and ingesting all of this food and medications and drinking water and all these different things that we're putting into our body. We have microbial checkpoints like stomach acid and bile acids and pancreatic enzymes, but then we have the small intestine that also brings in that element of protection. So we've probably all heard how the intestines contain the greatest number in diversity of immune compartments. So it's about 80%, we say, of our immune system is actually in the GI tract. And when we think about these different immune compartments, we can break them out into 4 different pieces. With the mesenteric lymph nodes, we have the galt. So the galt is part of the immune compartments of the intestine. We're going to break these out in a second. We have the lamina propria, which is a part of the intestines, and then we have the epithelium, so that single cell layer that we were just talking about a few slides back. So these are the 4 areas that help to build the immune system that's within our gut. So with Peyer's patches, again, we're still in the small intestine right now. We're talking about what's going on in the immune system there. These are our main immune cells, and they're in this lamina propria. So it's kind of this part of that mucosal layer, but it's just that area right underneath the epithelium. But that density starts to increase as we go towards the terminal ileum. So it's higher numbers in the small intestine, but again, that density increases. And essentially, its role is to identify any sort of antigen, a food antigen or a pathogen, and it helps to start to produce those antibodies, like specifically secretory IgA. Now, our numbers of these Peyer patches increase as we age to about the age of 25. But then after that, the numbers actually start to decrease. So again, if we think about as people age and some of the immune system dysregulation that can happen as we age and in older populations, part of it can be through just this decrease in these Peyer's patches. So this is why gut health, making sure that we're talking about, good microbial balance and reducing dysbiosis and mucosal balance and trying to do everything we can to preserve a really healthy intestinal epithelium, whether that be through supplements or through diet and lifestyle. Part of it is to kind of counter and support what we're naturally losing as we're aging over the age of 25, which are these the number of Peyer's patches that are present in the small intestine. And it does consist of various immune cells, so the B cells, T cells, macrophages, etc. So this is the kind of the overview. Let's talk about the anatomy, and then we'll talk about the function. So we have these Peyer's patches, which are underneath the epithelium, and then the little single cell layer, that epithelial layer that is covering the Peyer's patch. So it's still part of that entire intestinal epithelial lining, but it's just this one little section that's over the Peyer's patch. This is called the follicle-associated epithelium. So it's kind of a special section of the epithelium. And it contains these M cells. So M cells are these little pockets, and they are allowing this direct interaction with the immune cells. And it helps to also shuttle free and also bound secretory IgA antigens into the Peyer's patch. So it's sort of a gateway that then presents these antigens that are coming into these dendritic cells. There's also this called SED, or the subepithelial dome, which is where we have the dendritic cells, which are these sampling cells that are constantly sampling whatever is coming in through these M cells. So let's say we have a pathogen, or maybe there's a food antigen, or there's something that our body needs to deal with. We consume it, it's making its merry way through the small intestine. It comes into contact with these M cells. Secretory IgA gets involved. They then get plugged through and bound to the dendritic cells. And that's then when it starts that cascade of kind of the immune system figuring out what to do with that particular antigen or that pathogen. So here's the function. It's immune surveillance. So the steps are pathogen meets M cell, presents to dendritic cell. The lymphocytes or immune cells are activated, which amplifies the immune response, which then through these mesenteric lymph nodes, then carries out this whole kind of immune system regulation of dealing with whatever antigen it was that came in. So let's go back to talking about SIBO, or some sort of a condition that might disrupt the intestinal epithelial barrier, or maybe something that's disrupting the mucosal barrier. This is how, and when we talk about gut dysbiosis triggering immune dysregulation, or when we talk about leaky gut triggering immune dysregulation, it's basically this breakdown of these cells in these very specific parts of our intestines that are being compromised. Or let's say you have, if any of you have done biome effects consults, there are certain bacteria that we know could potentially degrade or interfere with secretory IgA. So let's say you have lost now, or not lost, but you have some compromise in the function of secretory IgA. You've kind of lost the signaling soldier who's there to essentially protect and regulate this immune response. So this is how, when we talk about gut health, it triggers in and factors into immune system dysregulation. So basically the bottom line here is that these Peyer's patches are keeping our intestinal bacteria and our intestinal flora at appropriate levels, and then also preventing any sort of infection. Now, in the very end of the the very lower part of the small intestine, the ileum, and as we get into the large intestine, we have what are called these isolated lymphoid follicles, which if you look at this compared to a Peyer's patch, it's a smaller, much smaller complex, but it is a major GALT in humans. So we do have a lot of these, and you can see in this diagram here, there's a couple of different types, but for your knowledge, it's essentially throughout the entirety of the colon and in the lower part of the small intestine. So this is the immune system component that's part of the large intestine. And so why is this all important? Why are we spending so much time talking about the immune system and epithelial barriers and cell function? Well, it all goes back to leaky gut, which I think we're all probably pretty familiar with the concept of leaky gut, because the barrier integrity of our intestines really depends on two things. We have tight junctions, and then we have the mucosal barrier. And so just to break it down here, again, I don't want to get too into the weeds because you're going to go over this a little bit more in future webinars, when they're going to talk a little bit more about the intestinal barrier system and leaky gut. But we have, the occludin and the claudin integral tight junction proteins, which are connecting the cells together. And then we have the zonula occludins, or the zonulin that's kind of within those intracellular proteins. And then we have the mucosal barrier, which again is made up of the mucin. So we have this mucus production, we have the antimicrobial peptides, we have the IgA, and then we have the tight junctions. So all of these things are coming together, the tight junctions, the goblet cell production, which goblet cells, just as a side note, if you have too much inflammation, but also if you don't have enough, so if you have to have a really nice, it's kind of like Goldilocks, you have to have kind of enough, and it's normal for us to have some level of inflammation. But we don't want to have too much, we don't want to have too little, because it's that little bit of inflammation that actually helps to stimulate that mucus production. If we have too much inflammation, so IBD, Crohn's, celiac, SIBO, it's actually going to affect the ability of those goblet cells to really secrete that mucus. So then that's when we start to get that mucus degradation and that mucus, suppression. And that's when we can start to get that breakdown. So it's important to think about goblet cell function. And we definitely have some interventions that we can talk through with that. So basically, disruption of any of this is going to lead to that enhanced intestinal permeability and can lead to systemic inflammation, which can lead to things like autoimmunity. So this is when we're talking about autoimmune, and when we think about the three pillars or the three legs of the stool, so to speak, of autoimmunity, it's having someone who has a genetic predisposition, something that runs in their family. An environmental trigger, whether that be the westernized diet, whether it be a medication, whether it be a pathogen that they come exposed to, and then leading to things like dysbiosis, leading to an intestinal epithelial barrier dysfunction, leading to a more inflammatory response, damaging intestinal tight junctions. It's that leaky gut syndrome that then is kind of that final straw that then leads to that autoimmune condition. So you can start with somebody who has maybe some genetic and environmental factors, but if things aren't dealt with, or if there's not interventions, it then progresses into this basically flipping of the switch to autoimmunity. And so, and it underlines a lot of other things. I mean, we can trace leaky gut syndrome back to all kinds of different conditions, whether it be mood disruptions, metabolic disorders, that type of thing. All right. So the gut-brain communication, again, I have just one slide on this. I'm not going to go too much into it just because you guys are going to hear more about it in future webinars. But I wanted to at least just do a little sprinkle for you, just kind of introduce it. So you've probably, if you've listened to any of our webinars, you've heard a lot about the gut-brain axis. Essentially, it is a complex. It's not just the vagus nerve, but it includes the vagus nerve along with our endocrine system, our HPA axis, our immune system, and our gut microbiome. And if we think about the enteric nervous system, what's fascinating is that it actually has more nerves than in the spine, and it produces more than 30 neurotransmitters. So we have quite the nervous system within our GI tract. And a lot of the hormones and the different peptides that our enteric nervous system releases can cross through that blood-brain barrier, and it can interact with the vagus nerve. And most of these signals coming between the vagus nerve and the brain and the gut, so that vagus nerve that connects our brain to our gut, most of the signals are actually going from the gut to the brain. About 90% are what are called afferent, meaning it's going from the GI tract to the brain or to the central nervous system, versus about 10% of signals which are going from the brain to the gut. So if we have gut disruption, then we know that there can be potentially some neurological or some brain inflammation, brain disruptions that can happen. And really the vagus nerve is responsible for regulating many, many different functions. So digestion, heart rate, respiratory rate, swallowing, sneezing, vomiting. Vagus, it comes from vagabond, so it's the kind of the wanderer. So if you look at the vagus nerve, you'll see that there's kind of two sides to it, and it sort of wanders and it connects to the various organs, and then it kind of feathers out in the digestive tract and connects to the various digestive organs. Just wanted to introduce that to you guys. Okay, so let's just take a pause. Let's take a breather, kind of think about all of what we discussed. And I would just be curious, feel free to talk about this in the chat. I'm curious to kind of hear what you guys say, but how would any of the information, maybe it's just one pearl that you learned, or maybe it's, kind of before we transition to the next part of this presentation, but how does this information support what you're maybe already doing in clinical practice? How does it maybe affect how you'll change your clinical practice? I'm just curious to hear. So feel free to pop into the chat any little pearls or little pearls of wisdom that you've taken away on how you'll maybe integrate this into your practice. All right, pause, break, switch gears. So now that we've talked about the anatomy of the intestines, let's talk about digestion, going from top to toe. And again, the reason I'm doing this is because when we are thinking about somebody who's struggling with some sort of digestive symptom, or just any symptom, it could be anything from depression to food reactions, to skin issues, to rashes, to whatever it might be. And if we think that there's a GI component, what's happening from the top all the way down through the bottom, we have to think about what's going on and where there's a breakdown in that function. So that's why I'm going to take you through just to kind of remind everybody what's going on in each stage. Because the more you understand each of those sections of the digestive tract, the more you can pinpoint and better pinpoint what you might need to do with that particular individual. So I'll start with the brain, which we don't always think about as a digestive organ, but we do have a cephalic phase of digestion, and that is completely 100% mediated by the vagus nerve. Because it's when we have the sight of food, the smell of food, the thought of food, we're starting to get hungry and you're starting to cook, your meal, and you start to have those wonderful flavors and smells, and you start to think about the food and the texture, and that's the cephalic phase. And that accounts for about 20% of your gastric secretions. So before you've even eaten anything, you have about 20% of your gastric secretions associated with a meal being secreted at that point. And so the questions would then be, what if you have someone who's eating mindlessly, or maybe they're not cooking, maybe they're not engaging in that process of eating and cooking their meals, or maybe they're just drinking their meals, or just grabbing a pre-made smoothie and slurping it back. They're not chewing anything, or maybe they have a pre-made protein shake. That's not going to be encouraging any sort of cephalic phase. What if they have a head injury, or they're stressed, or maybe maybe they're on a medication, or they have some sort of situation like major congestion or allergies that they're just not interested in food, or they have some nasal polyps. Those are all going to be things to think about that, they may have an issue with their cephalic phase of digestion. They may not be priming their digestive system very well in preparation for that food to come in. So, doing things like enzymes, or thinking about finding maybe lifestyle ways for them to start that process of that cephalic phase, maybe like smelling a lemon or something that can help them with that process. When we get down to the mouth, we have to think about chewing. I will admit, I am not the best when it comes to chewing sometimes, especially if I'm in between meetings or there's something going on, but we have to chew about 30 to 40 times to properly break down food before swallowing. And if we don't chew enough, then that can lead to a lot of the same symptoms that we think about with H. pylori or SIBO or some of these other digestive conditions, but sometimes it might just be that they're inhaling their food and they're just not chewing. Saliva production is important. So we have mucus, we have different enzymes that start to break down the starches and the fats, as well as we have lysozymes in there, which helps to break up bacteria. So again, having not enough saliva production, what's that going to do for your oral microbiome? And oral hygiene. So this is another thing that I always ask clients about is, what is their oral hygiene like? Because the oral cavity is our second largest microbiome after the gut. So we have the largest in our gut, and then we have the second largest in our mouth. So the two areas, kind of that are the two openings, is where we have our largest groupings of bacteria. And so what's happening in the mouth can affect what's happening in the gut. And this is just from an article that, and the references are there, that just shows some of the different species in the mouth that can be associated with various diseases or disease states. So again, if you're doing BioMFX, you may want to look at this, because if you see something like high Prevotella or maybe high Proteobacteria, ask them about their oral health history, ask them about their oral hygiene, because if you're seeing a lot of those things coming down from the mouth, getting into the gut, then there could be something going on with dysbiosis in the oral cavity, or there could be a breakdown in some of those antimicrobial digestive checkpoints, like this stomach acid or pancreatic enzymes or something along those lines. So just wanted to kind of tie that in. So these would be some other things that you'd want to look for in a client's history that could affect their oral hygiene or their oral health. TMJ, obviously they're not going to be able to chew very well if they have TMJ. Certain medications, if they have like Sjogren's or if they have lupus, those are associated with low saliva production. So they may need some support for their saliva production, or maybe some of those saliva substitutes that are available. Diabetes, definitely. People with diabetes have more of a predisposition for oral candidiasis because of having hyperglycemia. And ask them, are they chewing a lot, or are they having to chew a lot, or are they maybe always the last to finish their meal? That might be an issue with maybe their chewing. And so looking at those things. So moving to the esophagus, we have our vagus nerve, which again is going to be innervating the esophagus. And then we have the lower esophageal sphincter, which is just that bundle of smooth muscles that help with that relaxation and contraction of that, kind of to control that reflux of gastric contents into the esophagus. And so if someone's, again, having a lot of GERD or reflux, then, first ask them about any of the foods that could potentially, or food ingredients that could potentially cause excessive relaxation. These are common ones that we're probably all very familiar with. But also, too, some things like chronic coughing or burping. Burping may not be a low stomach acid problem. Maybe it's an issue with the lower esophageal sphincter. And then mast cell activation. Mast cell activation can cause issues with the function of this lower esophageal sphincter. So always kind of going back and asking the why. But this is why some people with mast cell can have more of that refluxy type presentation, or they can have other issues, because it can certainly cause inflammation and affect the ability of the sphincter to work properly. So here are some factors that we have built in to our digestive system that help to keep microbes in check. Oops, didn't mean to go to that. So we have our gastric acid, which is obviously very acidic. So it typically will eliminate, or it'll kill most of the bacteria that are coming through from whatever we ingest. We have pancreatic enzymes, which are also antimicrobial, bile acids, which have an antimicrobial effect, and then our migrating motor complex, which helps to kind of sweep that bacteria and keep it from overgrowing. And then we have the ileocecal valve, which helps to prevent that backflow from the colon to the small intestine. And then our mucosal immune system, which we've talked about. So this is essentially, I would say, probably one of the most important slides to kind of note or star or pay attention to, because you can pretty much trace back almost, I won't say all, but almost most of the digestive insufficiencies or the digestive symptoms back to something happening here, whether it be somebody who has low stomach acid, maybe they have poor pancreatic output, maybe they don't have a gallbladder, or maybe they have something going on with their liver, maybe they have bile sludge, and so they're not secreting their bile very well. Maybe they have something affecting their small bowel motility. Maybe there's something going on with the immune system. So maybe they have a lot of intestinal inflammation. So their immune system, like we've talked about the whole first part of this presentation, can't manage those microbes. So this essentially can kind of help you to say, where do I want to focus my attention and where do I maybe need to investigate a little bit more to figure out that reason for why they have those imbalances. All right, so the stomach, like I mentioned, it has its own epithelial line and it has only mucus cells for obvious reasons. We have a lot of acid in our stomach. We need to have, sufficient mucus production to protect that epithelial layer so that we're not getting a bunch of ulcers. And we have these gastric glands or these gastric pits that are basically this entry to the gastric gland, which is where we're getting that gastric juice production. And gastrin is the hormone that stimulates that gastric juice. So that's what's stimulating the gastric juice production. So then we have these parietal cells, which are making the stomach acid. They also make intrinsic factors. So it's helping to keep that stomach at a proper pH and helping to kill off the bacteria. And then the intrinsic factor is there to help with the absorption of B12 into the small intestines. So again, if people have an issue with H. pylori, or they have gastritis, or they have something going on that's affecting a lot of stomach inflammation, these are some of the things that are gonna suffer. Now, the chief cells, they secrete pepsinogen, which is the inactive pepsin, which helps to start breaking down our proteins. So people who have a problem with digesting or breaking down protein, or maybe again, if you're doing BioMFX and you see someone who has a lot of proteolytic fermentation byproducts, so they have a lot of protein fermentation, there is probably something going on with these cells or something going on with the small or the stomach acid secretion, and that's triggering some of those higher protein byproducts. Now, there's a gastric phase of digestion, which is about 3 to 4 hours. And this essentially is when we've ingested food enters the stomach distends. That distension then triggers a hormone or a neurotransmitter, acetylcholine, which then triggers that secretion of the gastric juice. So again, think about that cephalic phase, that kind of beginning part of the cephalic phase and leading into this gastric phase. And then gastrin, which is what's triggering that stomach acid release as well, is going to be activated by those proteins, caffeine. So this is why sometimes when people are drinking a lot of coffee or they have a lot of caffeine, it might cause more stomach issues because it might be stimulating more of this gastrin. And then that's when we get also the protein digestion as well, as we kind of talked about. So these would be some signs of low stomach acid. A lot of them are pretty self-explanatory, but also here are some signs of if somebody has ulcers or they have maybe some gastritis. So these would be just a couple, again, more for your reference to kind of look at to see what may be going on with your client. Now, we've talked a lot about the small intestine from an immune system standpoint, but again, just to kind of go through real quickly some of the functions. This is where most of your chemical digestion is occurring in the small intestine, whereas mostly mechanical, it's like in the mouth. But again, that chyme is coming from the stomach into the duodenum. It mixes with a bunch of pancreatic juices and some bile, and it makes its merry way to go through for that absorption and that nutrient assimilation. And it can take about 5 hours for a single meal to be processed through that entirety of the small intestine. And it's really, though, coordinating with our other ancillary organs, the gallbladder, the stomach, and the pancreas to really absorb about 95% of our nutrition. So the duodenum is that first part. It's a little bit more acidic than the lower parts of the intestine because that's where the stomach is dumping its digestive juices. And so it's a little bit more acidic, but we also have a lot of mineral absorption. I put some of the vitamins and minerals there for you. So you can kind of think about if someone's deficient in any of these, what might be going on with their small intestine. The jejunum is that second part. There's some mechanical digestion that occurs there, and it's where you have folate absorption and as well as you continue to absorb a lot of your vitamins and minerals. And then in the ileum, which is that third part of the small intestine, and this is where most of the food is going to spend its time when it's going through the small intestine. A lot of it's going to spend its time in the ileum. And this is where you have that ileocecal valve that separates the colon from the ileum, that's preventing that backflow from the colonic contents. And there's also a lot of B12 and bile salt absorption that's going to happen here. This is the main site of where that's going to be going on. So what if somebody has their ileum removed? You have to think about B12 and bile salt absorption. Or if they have a lot of inflammation, like again, a Crohn's or a celiac or a SIBO, be thinking about B12 deficiencies or issues with their bile salt absorption. The pancreas has two functions. It has both endocrine and exocrine function. So you have your things like insulin and glucagon, which are secreted, but really 80% of the pancreas is an exocrine in function, which is more of the enzymatic function of the pancreas. And so again, food is passing from the stomach into the duodenum. That's going to trigger this release of CCK, which then tells the pancreas to release bicarbonate, because we don't want this really acidic chyme going through the intestines. That would hurt. That would not be comfortable. So this is where we have that alkalinity of the bicarbonate coming in. But the other reason is because most of our digestive enzymes function at a more alkaline environment. They can't function if it's too acidic. So you have to have the right pH in your intestines to then signal for these enzymes for them to not only be released, but also for them to work appropriately. So if your pH is thrown off because you're not getting proper signals from the stomach, pancreas, and from the small intestine, your enzymes aren't going to be as efficient, or they may not be able to work. So CCK, as I mentioned, it's secreted by the small intestine. It also releases bile into the intestine, as well as the bicarbonate, and then secretin, which is secreted by the duodenum to stimulate that from the pancreas. And then we have amylase, lipase, and trypsin. So we break down things like starches, triglycerides, or fats and proteins. Oops. So what would you want to look for? So anybody having difficulty digesting fiber, which could also be a SIBO issue, but it could also be a pancreatic enzyme issue, nausea, vomiting, left side pain under the rib cage, those types of things. Definitely a history of diabetes. Again, I'm not going to go through all of these just for time, but here is a list for you of what you might want to be considering if your client's checking a lot of these boxes. So with the liver and gallbladder, we have this whole biliary system that's connecting the liver with the gallbladder and the pancreas to then dump into the small intestine. And when we talk about bile, it's kind of the middle child. A lot of people think about stomach acid and a lot of people think about enzymes, but sometimes bile and bile function gets kind of lost in some of the clinical side of things. And so I think it's something that is underutilized and it's not always addressed, but it is produced, or it's made up of cholesterol, bilirubin, bile salts, and phospholipids. It's a huge elimination route for toxins. So we excrete a lot of our toxins and our hormones, and also cholesterol. Most of our cholesterol is actually excreted through our bile. So if people are having cholesterol elevations, or if their cholesterol is going up, yes, it can be diet and lifestyle, but it could also be if they don't have good bile flow and function. Obviously, it emulsifies fats and helps with the fat absorption, but it can also have an immune system component. So it can stimulate the innate immune system, can help with that IgA excretion. And it also can deliver vitamin D from the liver to the intestine, as well as things like B12, folate, and B6. And it's a signaling molecule. Bile acids bind to receptors throughout our body that regulate then glucose and energy metabolism. So sometimes, when people are having more cardiometabolic problems, if they have poor bile flow and function, it's because they're missing out on these signals that are binding these receptors to then trigger this cascade of other signals that can regulate some of that cardiometabolic function. So again, this is just a review of: we eat fat, CCK is released, the liver, gallbladder release bile, and the pancreas releases enzymes. That's just the breakdown of the digestive piece. So a few pearls here for you. So our gallbladder emptying starts when the stomach begins to empty. So the stomach and the gallbladder sort of have this coordinated emptying that happens. So what if we have gastroparesis? What if we have a sluggish gallbladder? You're going to miss out on that coordination. So think about somebody who has diabetes. They might have that gastroparesis, or someone who is really stressed and their gastric emptying is really fast or maybe really slow, depending on how chronically stressed they've been. That could tie into that coordination of how well the gallbladder can then empty. The other thing is that response of the gallbladder to CCK to release that bile is calcium dependent. So what if someone's on calcium channel blockers like Norvasc? That can potentially decrease CCK response by 80%. So maybe their gallbladder dysfunction is related to some sort of calcium imbalance that they have. The other thing too is the vagus nerve. So if somebody's very, very stressed, or if they have a vagus nerve that's in a constant sympathetic state, they can't really efficiently or as effectively have that appropriate gallbladder contraction. So if someone's stressed when they're eating, they're just not going to do very well with their digestion. So this is where, again, that mindfulness, the breathing, I love all of the music you guys were talking about in the meditation at the beginning of the webinar, playing that before a meal, breathing before a meal, just sitting for the meal and not eating on the go, all of those things. And then here's a list of some potential signs of inadequate function. So again, not going to go through this, but just wanted to kind of summarize for you a few of the different hormones, a few of the different digestive hormones, where they're produced, what they're stimulated by, and what their action is, just as a summary slide for you to kind of refer back to. All right, so we'll kind of wrap it up with the large intestine, which again, we've talked about this, but primarily is absorbing water and electrolytes. And again, by the time things have gotten to the intestine, the large intestine, about 90% of that water has been absorbed. And so with the ascending colon, you're absorbing any remaining water and other nutrients. You're starting to solidify that stool. The descending colon is going to store that stool. That's going to eventually empty it out into the rectum. And then the sigmoid colon, which is this little section here, is going to cause that contraction to start to increase the pressure to actually have that stool go into the rectum for elimination. And again, the things like vitamin K and a lot of our B vitamins are produced by our colonic bacteria, and they about 20% of what we make, 25 to maybe up to 50% I've seen of these vitamins that our bacteria are making are then absorbed into the bloodstream. And so there is this idea or this theory around kind of this compensation that if someone's deficient in intake of these vitamins, that the colon kind of the bacteria sort of compensate by increasing their production of these vitamins to then minimize that vitamin disparity. So you'll see some of these vitamins listed on bioeffects tests. And so you can kind of look to see, well, what is the microbiome actually contributing? Butyrate is a big one. We've talked about the ileocecal valve. Butyrate is another, very important short chain fatty acid that helps with maintaining that repair of the colonocytes and energy production. So again, low butyrate, if you're seeing that on stool testing, then you're going to be looking at, risk factors for things like colitis. It's associated with things like ulcerative colitis and other types of colitis, IBD, that type of thing. Now, and I always love to do a transit time test, even if you have a person, a client who's having a bowel movement every day, or they're reporting they're having a bowel movement every day. It doesn't mean that they're fully moving their bowels every day. So I like to do a sesame seed test, which is where you basically eat, a tablespoon or two of some sesame seeds. You could use corn, you could use beets, you could use charcoal. Basically, you're using anything that is going to come out in the stool, and you'd be able to see it. If you're seeing, and so they time when they eat it, and if they see that that thing is coming out less than 12 hours, then they probably have really fast transit and they're not absorbing their nutrients very well. The ideal transit time would be about 12 to 24 hours. So, so that's why I say, like, every morning have a bowel movement, or maybe twice in a day, like a morning and an evening bowel movement. That's really what we're looking for. If you have somebody who maybe they've had a bowel movement, but if they've eaten sesame seeds on Saturday midday and they don't see it coming out until Monday afternoon, even if they've had a bowel movement, and I've had people where this has happened, they still have slow transit time because it's sitting in the bowels over 24 hours. And that waste can be, sitting too long in the colon. So I like having people do transit time tests regardless of what they're reporting in terms of frequency of their bowel movements, because sometimes you might be surprised with how long it actually takes them to move that food through their intestines. And then again, detoxified hormones like estrogen are secreted in the stool, and that will be talked about later in your future webinars. So these would be some other large intestinal signs of inadequate function, and also some things to look for in a client's history. So just basically in summary, we have a pretty fabulous digestive anatomy and physiology that works for us. It's always trying to work for us. So if we have symptoms that are working against us, it's because the body's trying to compensate for something. There's some sort of a signal that is crossed, or there's a dysfunction going on. And so we have to understand that hierarchy and the flow of the digestive system and rely on those previous signals for things to work the way that they should. So as you progress through these foundation webinars, you'll start to connect more of those dots between the foundational concepts. So these are the future upcoming educational opportunities. We have our March and April foundation series with Kara and Dr. Sarah. And then we have coming up later this month, the next BioMFX installment with Dr. Sarah. And then I'll be presenting in March on the upper digestive tract with BioMFX. And then we do have our mini keynotes on March 12th at, I believe it's 2 Central Time, but Angelica, you can correct me on that. So March 12th will be mini keynotes, and then we have our other webinars as part of the series coming up. And with that, am I on time? I think we have maybe, I don't know, 10 minutes or so we can do for Q&A, 5 or 10 minutes. But I'm also curious, just if you have a chance to drop in, what is one new thing you learned, or maybe you were refreshed and relearned today in this webinar? I'm always curious to hear about just what you've learned. And yes, I see people asking about the slides. The slides will be sent out as well. So awesome. Thank you so much. Yeah, just looking through some of these. All right, and then we can go into some Q&A. Let's see here. Is there a probiotic that you recommend for improving the oral microbiome? There's not really any particular one that I recommend. There are a few on the market. Really, it's just about making sure that your oral hygiene, you're brushing appropriately, you're using the proper types of toothpastes that aren't filled with things like fluoride, not a bunch of chemicals and SLS and that type of thing. So it's really just about flossing. Things like sugary foods can be problematic for the oral microbiome. Any comments or recommendations on alpha-gal? So alpha-gal is like alpha-gal syndrome when someone who has like this, whether it be a beef allergy or this basically allergic reaction to like protein containing foods. I mean, I don't know in terms of like comments or recommendations, there's a lot of different, food avoidance. For some, it can sometimes go into things like supplements, like definitely you would want to do any sort of IgG type of product if somebody has an alpha-gal syndrome because of the bovine proteins. It's really about management of food. And even for some people who have alpha-gal, it's not necessarily that all of the foods that are typical to trigger alpha-gal. Like I was saying, some of the food additives, you don't really have to worry as much with those. But for some people, if it's severe enough, you may need to. So what are the calcium channel blockers? So Norvasc is one. You can, and there's a few others. You'd have to kind of look up whatever your patient's on to see if it's a calcium channel blocker. But Norvasc would be one. What butyrate products are recommended? What to look for in various brands formulations? Are these helpful? So you can, butyrate, you can do it exogenously. And for some people like IBD or colitis, it's helpful, or they might need butyrate supplementation. But you want to try to get them to a point where they're actually producing their own endogenous butyrate. So you can use things like there's a brand, Tributyrin, that's a really good brand that you can use if you're giving it exogenously. But basically what the research shows is that over time, like you're just kind of putting a Band-Aid on an underlying condition. You want to try to get it to where you've got your own endogenous microbiome making that butyrate. So that's much more effective. It's going to have much more of an effective long-term solution for that person. So, gut reconditioning, short-chain fatty acid support through things like specific fibers. That's what you'd want to transition to, but Tributyrin is a good brand of butyrate if you're going to do it exogenously. How to improve saliva production? It depends on the reason. You can do things like a lemon, like having smell or taste a lemon before a meal. That can help to stimulate saliva production. There are a few oral saliva substitutes on the market, like Zilly Melts and Aqua. There's an Aqua, not Aqua True, Aquasol. Zilly melts, there's Oasis is one. So there's a few different saliva substitutes that you can also, if somebody has something like a Sjogren's or lupus where they can't make that saliva, the lemon may not be enough for them, and they might have to have something else just to help them with that chewing process so that they're not, having a compromise with their chewing function. Food combinations, possible to talk a little bit about that, protein and fruits, good or bad? Yeah, I don't really, food combinations, I know some people anecdotally really, believe in that, and yeah.
I wanted to hop in. She added another question that said she meant in terms of digestion time, alkaline versus acidic environment. So combining foods and how they digest it.
Yeah, again, there are people who really swear by this, and they've seen good outcomes. So I'm not here to say that this is not, if you are using it with your clients and you're seeing good outcomes, great, keep doing it. Your digestive system is more intelligent than that. It's not, it's not like we've come up with this idea that, oh, this is our new way of food combining and we're teaching our digestive system. Your digestive system is equipped to eat, like to just deal with some of these things that are coming in whatever combination it is. Because if you think about when we were off in the, in the wild, having to scavenge for food and eat protein, I mean, we had whatever we could find, whatever mix. So I don't really feel like that you have to worry as much with that food combining for, alkaline acid environment. We have a much more intelligent digestive system. How long can you keep a patient on HU58 and IgG2000? Really, as long as you feel like you need to. There's not a safety issue with those two things. So as long as they're, having benefit. I mean, I'd wonder why they're on the IgG2000 and HU58. So you'd want to work on that root cause of whether it's diarrhea or maybe you're trying to get rid of some pathogens. You still want to work on root cause level, but yeah, you can keep them on as long as you feel like you need to. How does removal of the gallbladder impact digestion? So a few different ways. One is when someone has had their gallbladder removed, they will probably have been told by their doctor that their body will still make bile, and that is true. It will. However, the bile that our liver makes is a totally different composition than what the bile is in our gallbladder, because the gallbladder concentrates that bile. So the bile salt content is much higher in the gallbladder. The water content is much lower in the gallbladder. It's where we're also creating that conjugation to make our secondary bile acids, which is what we need to make to actually digest our fats. In the liver, we have primary bile acids. And then once they go to the liver, from the liver to the gallbladder, we get our secondary bile acids through that conjugation with taurine and glycine. So yes, we still make bile in the liver, but it's a really very different electrolyte composition. So if someone doesn't have a gallbladder, at least initially, there is going to be some compromise in their digestive function. And also too, there are studies that have shown that the removal of the gallbladder, it changes our microbiome, and it also can delay oral fecal transit time. So things like SIBO, about 2 years after gallbladder removal is when you are most likely to see the presentation of SIBO. There are studies that have shown that it's not necessarily immediately, but it's kind of over time as that oral fecal transit time has slowed down and things have kind of slowed down. It can affect and lead to the development of SIBO. So it is a risk factor. So those are a few different ways that it can affect digestion. And some people need ox bile or bile salt supplementation initially, and they might need it for longer term. Some people eventually could get off of bile salts, but they definitely will probably need it at least for the first, initial part of having that gallbladder removed. Do we know if caffeine tomatoes for mom increases gastrin in a nursing baby? I don't know. That's a good question. I would have to research that. So that's a good question. Now, I'm not sure if it would. Maybe somebody else here does know, but I don't know if it would necessarily increase that in a nursing baby. I mean, I guess it could. But again, a baby's digestive system is not completely, like it's still kind of developing. So there's there's some different things that are going on in a baby's digestive tract than what's happening in an adult. So, but that's a very interesting question. Are there tests for a good diverse oral mucosa? There are a couple of, I think there's at least one that I know of test on the market for an oral mucosal, like microbiome composition. I want to say there's two, but I know there's one, it's called Bristol. How do you put increase transit? Maybe how do you increase transit time? You can use things like prokinetics, like for SIBO is used a lot, which can help with coordination of the gastric emptying and the small intestinal transit time. Things like laxatives, obviously, but that's going to work more on the colon. So a prokinetic will work on the stomach and the small intestine. The laxative will work on the colon. So that kind of works in two different parts of the intestinal tract. You can do vagal nerve stimulation. I mean, it depends on why they have slow transit time. Do they have methane, SIBO? You'd want to work on the methane. So it would depend on why they have that slow transit time in the first place. I've heard you can use H250 as a mount, for instance, to help with oral bacteria. Yeah, I think we've had some anecdotal practitioners who use that, and it's been helpful. Can arrhythmia be caused by an impaired LES? Yes, it can. Can you use TUDCA for gallbladder removal? You can. Yeah, TUDCA works a little bit differently from bile salts, but it can, it actually does help to stimulate some bile production. So TUDCA can be a good additive as well. So, all right. How did we do on time? Okay, 10 minutes. So we went about 10 minutes over, but I got all of your questions answered. So thank you so much for joining, and thank you for those of you who have hung in there. We really appreciate you joining us live, and look forward to you joining us for our next future microbiome webinars. And I wish you all well and hope you have a wonderful rest of the week.